antibody structure and functions. Antibodies are protein molecules produced by plasma cell as a result of interaction between antigen-sensitive B lymphocytes and specific antigens. They have property of combining specifically with that antigen, hastening their destruction or elimination. B cell receptors are special antigen receptor on the surface of B cells, but these proteins are not restricted to B cell surface. Once the immune response is triggered, they are shed into surrounding fluid. These soluble forms of BCRs are known as antibodies. Edelman and Porter discovered the structure of antibody in the year 1959. They won Nobel Prize in 1972. They also did amino acid sequencing of heavy and light chains. They used 2-mercaptoethanol that irreversibly cleave disulfide bonds without affecting peptide bonds. Structure of antibodies. These are glycoproteins, heterodimers with 4 polypeptide chains, that is 2 heavy chains and 2 light chains. These are linked by disulfide bonds. When an antibody molecule is treated with pepsin, FAB2 fragment is generated that can bind to antigen and cause visible reaction, that is agglutination or precipitation. The Fc fragment is digested into multiple fragments, hence not recovered. When an antibody molecule is treated with papain, 2 FAB fragment is generated that can bind to antigen and cause visible reaction, that is agglutination or precipitation. An Fc fragment generated is also known as fragment crystallizable, as it crystallizes during cold storage. An antibody molecule consists of 4 polypeptide chains, that is 2 light chains and 2 heavy chains. Both types of chains consist of constant region and variable region. The variable region in light chains further consists of low variable region and hypervariable region. A monomeric immunoglobulin G molecule consists of 3 globular regions, that is 2 FAB and 1 FC, linked by a flexible hinge region. Each of these globular regions is made up of paired domain. Thus, FAB of each has VH linked with VL and CH1 linked with constant light chain. The FC of each has 2 to 3 paired domain depending upon the type of immunoglobulin class, that is CH2 linked with CH2 and CH3 linked with CH3 in case of IgA, IgG and IgT, and in IgE and IgM, that is additional CH4 domain linked with CH4. In FAB globular region, there is groove located between 2 variable domains, that is VH and VL. The peptides of complementarity determining regions lie in these grooves, hence show great variation in its shape. The groove forms antigen binding site known as CDR or paratope. CDRs from both light and heavy chains contribute to binding of antigens, although heavy chains contribute the most to process. Because immunoglobulins are bilaterally identical, it has 2 identical antigen binding sites, thus able to bind 2 identical epitopes together. This principle is applied in case of agglutination and precipitation. Each constant homologous region in both light and heavy chain possesses a single intra-chain disulfide bonds. These bond folds chain into a loop. Similar loops are also observed in variable region where they bring CDRs together. This loop formation due to presence of intra-chain disulfide bond is known as domain. There are total 12 domain, that is 8 in heavy chain and 8 in light chain. Hinge region is a proline and cysteine rich area situated between CH1 and CH2 domain at 216 to 231 amino acids in immunoglobulin molecule. It is also known as universal joint. It provides flexibility to antibody molecule. It is also vulnerable to cleavage by proteases. They are unique to immunoglobulin classes and subclasses. Interchain disulfide bond binds heavy chain to light chain. Immunoglobulin classes. There are 5 main classes of immunoglobulins, that is IgG, IgM, IgA, IgE and IgD, some of which include number of distinct subclasses. Each class has its own properties and functions determined by the structural variations of their heavy chains. There are 2 basic kinds of light chains called lambda and kappa chains, either of which can be associated with any of the heavy chain classes. IgG antibody. It is predominant immunoglobulin in serum, mainly responsible for systemic defense. Sedimentation coefficient is 7s, have molecular weight of 180 kilodalton, has typical B cell receptor structure formed by plasma cell of spleen, bone marrow and lymph node. They have 2 identical light chains, either kappa or lambda, and 2 gamma heavy chains. Subclasses include IgG1, IgG2, IgG3 and IgG4. It is smallest in size, so can escape through blood vessels more easily and participate in defense of tissue spaces and body surfaces. Can opsonize, precipitate and agglutinate antigens. Can activate complement system, but only if sufficient molecules of immunoglobulins have accumulated in correct configuration on antigen surface. IgG4 may bind to mast cells and participate in type 1 hypersensitivity reaction. IgM antibody is made and secreted by plasma cell in spleen, lymph node and bone marrow. It is present in second highest concentration in blood serum. It is present on B cell surface as monomer and its secretory form is pentameric in nature. It is the first antibody secreted during primary immune response to antigen and first antibody seen in ontogeny and in neonates. They have 2 kappa or lambda light chains and 2 microchains, that is heavy chains. A small cysteine and asparagine rich polypeptide known as J chain binds 2 units to complete the circle. J chain is synthesized by plasma cells that produce IgM. IgM antibody contains additional CH4 domain, more efficient than IgG as complement activation, opsonization, neutralization of viruses and agglutination, but lacks specificity. There are 10 potential antigen binding sites, but only 5 to 7 are functionally active due to steric hindrance. IgA antibody is a carbohydrate-rich antibody synthesized in the wall of intestinal tract, respiratory tract, urinary system, skin and mammary glands. These are secreted by plasma cells mainly located just under the body surfaces, found as dimer, trimer or monomer. Molecular weight as monomer is 150 kilodalton and up to 360 kilodalton as polymer. The secretory form is mainly dimer and trimer. They have 2 subclasses as IgA1 and IgA2. IgA have a typical 4-chain structure, that is 2 kappa or lambda light chains and 2 alpha heavy chains. Dimeric IgA is linked by J chain at Fc region of both IgA monomers. It is transported through intestinal epithelial cell bound to 72 kilodalton receptor known as polymeric Ig receptor or secretory component. It binds covalently to IgA dimer to form complex structure known as secretory IgA. Secretory component protects IgA from digestion by its proteolytic gastrointestinal enzymes. It is major immunoglobulin external secretion of non-ruminants. Protects mucosal membranes against microbial invasion. It cannot activate complement and can't act as opsonine but can agglutinate particulate antigen and neutralizes virus. It prevents adherence of antigen to body surfaces. It is unique as it can act intracellularly. IgE antibodies, also known as homocytotrophic immunoglobulin or cytophilic immunoglobulin, reagenic antibody or cytotropic antibody. It is of typical Y shape, also contain an additional constant domain, that is CH4, consists of 2 epsilon heavy chains and 2 kappa or lambda light chains, have 7s sedimentation coefficient and molecular weight is 170 kilodalton, has shortest half-life and fairly unstable as can readily destroyed by mild heat treatment. IgE acts as signal transducing molecule. It is found mostly bound to receptors on mast cell and basophils. When binds to these molecules, triggers rapid release of inflammatory agent from these cells, resulting in acute inflammation. IgE mediates type 1 hypersensitivity reaction. Also, it is responsible for immune response to many helminthic infestations. IgD is primarily a B cell receptor found mainly on the surface of B lymphocytes. It is usually co-expressed with another cell surface antibody called IgM, consists of 2 delta heavy chains and 2 kappa or lambda light chains, have 7s sedimentation coefficient and molecular weight is 170 kilodalton. IgD have long exposed hinge region that lacks disulfide bond, hence usually susceptible to proteolytic enzymes generated and readily destroyed on mild heat treatment. It is found very low levels in serum and does not activate the complement pathway. IgD function is unclear, but may act as a B cell antigen receptor and play a role in B cell development and activation. Thanks for watching and don't forget to subscribe and hit the notification bell to remain updated for more such videos.